Okay, thank you very much, Deborah. So it's a pleasure to be here on behalf of our CPTR Preclinical and Clinical Sciences Working Group. I'm going to give you sort of a progress report or an update on a topic that I talked about last year, and that's the effort, as Deborah mentioned, to look at the sterilizing mouse model and its predictive accuracy. So I'm not going to have any great results. This is really just talking about process up to this point, and hopefully within the next year we can come back with some real results. So Deborah showed this, and I think it just to reemphasize what we're trying to do, and that's to try and find new tools and new approaches using existing tools to close that gap, this translational gap between early preclinical and preclinical and early clinical studies and subsequent phase 3 trials, with the goal of trying to reduce the risk of late-stage attrition, meaning getting to costly and lengthy phase 3 trials with regimens that aren't going to achieve our goals. You know, I'm going to focus, of course, on preclinical models here, and the real question there is which of these models, used in what ways, can best inform the designs of clinical trials, best inform these critical decisions that are made along this development path. And so, you know, I think I've just sort of encapsulated the mission of this working group, which is to develop new tools, validate existing tools to the extent that we can, and approaches using those tools to address these important preclinical issues. And as Deborah mentioned, as a group, we started a couple years back with this landscape analysis where we really tried to go back, investigate models that were being used in the preclinical space, look at the data that have been generated using those models, look at how they might translate to clinical, and try and get ourselves a little bit further down the road towards qualified or validated models that we could begin to rely on. And so in early, as you've already heard from Deborah, the hollow fiber system stood out during that landscape analysis as a tool for which there was a lot of data that were very well suited to this sort of analysis, lots of qualitative data that were appropriate to this kind of predictive accuracy analysis. You know, so that was, I think, the earliest success here, which was the EMA's qualification opinion on this, and the endorsement by FDA as well on this tool as an additional and complementary tool to existing methodologies. Importantly, paving the way to using a tool like this in regulatory submissions, and Deborah has emphasized where exactly this tool, how it was presented in terms of the context of use for its utility in decision making and regulatory applications. So, but it was very clear from our interactions with EMA and FDA during that process that the hollow fiber system should be viewed, at least at this point, as an additional and complementary tool, and that it is not meant to replace in vivo models. So that still left room, clearly, for similar evaluations of in vivo models and their predictive accuracy. And I just lifted this quote here from the FDA article that accompanied the hollow fiber data publication in CID now 2 years ago, but really emphasizing, I think, the potential value of in vivo models above and beyond what aspects we might be getting from a hollow fiber system. And I think, you know, at least at this point in time, it's very reasonable to hypothesize that in vivo models have the potential to better reflect the phenotypic heterogeneity that one sees in vivo settings, heterogeneity in bacterial metabolism, bacterial responses to antimicrobial agents, that are reflective of interaction with the host and development of specific host pathology, for example. And also thinking about infections and TB lesions as in a three-dimensional sense. And so we clearly rely on penetration of drugs into a three-dimensional structure and again, potential differences in bacterial heterogeneity that are created by that three-dimensional structure. And that's something that's clearly easier to come by in the in vivo models. So we really look at these as I said, complementary models, each having their own advantages, but each having limitations. And so the key question that we're faced with is really how to take the variety of in vivo models that are available and have been used, none of which one might say have really been held to a predictive accuracy analysis with the same rigor as that was applied to the hollow fiber system. And so for similar reasons as the hollow fiber system was chosen, as we moved to thinking about an appropriate in vivo model to look at, we chose the sterilizing mouse model, largely because this was a model for which there was the greatest amount of existing data that correlated evaluation of specific drug regimens in the preclinical setting and for which there were subsequent clinical evaluations on which to make a comparison and judge the predictive accuracy. So I included this really just to make sure we're all sort of understanding what we're talking about when we talk about the mouse model of sterilizing activity and to show at least an example of how this model is often placed in drug development programs. So it typically follows single drug optimization in terms of PK/PD and dose optimization. Initial combination studies looking at new drug combinations often initially use shorter durations and more rapid endpoints like change in CFU counts over time on treatment. And then for regimens that show promise in such analyses, then going on and looking at long-term treatment durations and looking at relapse after completion of treatment as an endpoint is really the measure that we're talking about when we talk about a sterilizing activity model. So in our most commonly used model, this is initiation of treatment on day 0 when the bacterial burden is maybe 8 logs or so in the lungs, and then stopping treatment at various defined treatment endpoints and leaving mice untreated for 3 or even 6 months, and then coming back and sampling those mice at the end of this follow-up period to see which ones retain viable bacteria in the lungs, and we generally term that a relapse. So we're really looking at whether we have eradicated organisms that are capable of relapsing after the end of treatment or causing relapse after the end of treatment. So the rationale for choosing this model, again, have to do with the amount of data available on regimens that are in clinical trials, as well as the past and present role that this model has played in regimen development and informing clinical trials. So this one can go back to studies beginning in the 1950s where this model has been used to forecast the treatment shortening potential of rifampin and pyrazinamide as they were incorporated into the first-line regimen. Again, because we're looking at sort of the clearance of the last persisting bacteria in these animals, we feel that this is the closest correlate to the current phase 3 endpoint of relapse after treatment completion. Importantly, looking at this model at this stage in no way precludes looking at other models, Kramnik mice, marmosets, rabbits, you name it. One objective here was simply to establish a methodology and a precedent for how one might try and examine the evidence base and develop support for a particular model. So the aim of this particular approach was to try and quantify the predictive accuracy of this TB efficacy model for its ability to rank order regimens by efficacy and to estimate the effective treatment duration by evaluating the difference in the time that it takes to reach a certain degree, a certain relapse outcome. So to reach the time that, compared to a comparator, a standard regimen, how much treatment is necessary to reach the same proportion of mice relapsing, whether it's 50% relapsing or 0% relapsing. So we're looking at differences between the standard and the test regimens. And so, you know, I think, again, this is space that we're looking at in animals that was really not looked at in terms of the hollow fiber system. We haven't looked at sterilizing hollow fiber systems and its predictive accuracy for duration of treatment with combinations, although that work is clearly ongoing. So we assembled a group of content experts. They're all listed here from a variety of areas, academics, regulatory agencies, and various sponsor groups. This schematic just sort of defines the work plan. So Deborah's talked about context of use. What's the proposed applicability of this model? So we've developed a context of use statement. We've gone and created a data inventory. We've pulled together, this is all largely within the last year, pulled together data that from the whole of mouse studies that have clinical data that are also available for comparison of the same regimens. We've developed a proposed statistical analysis plan for the data, and we're fully expecting, in fact, we know that there will be gaps in the amount of data and the type of data that will, to some extent, perhaps compromise our ability to do the statistical analysis, but it's important that we identify those so that we can look at opportunities for future research and develop an agenda in that sense. But what we also wanted to emphasize with this figure is the iterative nature of this process. So this is a process that can keep going and going, and another example of how looking at the model can inform the utility of the model, and the potential utility of the model, and the types of data that may be needed to even extend its utility. So this is just describing now our current context of use. We've developed several different scenarios, but the first effort is really going to be to look at the ability of this measure, again, differences in treatment duration needed to reach the same relapse outcome as a way of rank ordering regimens. So we're proposing that this will be an additional and complementary tool to inform regimen selection to maximize sterilizing activity that'll be useful for regulatory submissions, and that these data, again, emphasizing relapse as the endpoint, will be used to calculate treatment effect sizes that'll then be used to rank order regimens and to estimate the clinical treatment duration as the initial analysis. The data inventory here I've listed. It's maybe difficult for a number of you to read. We're focusing first on data generated in the BALB-C or other commonly used mouse models. We are currently not, although we are intending to in the next steps, planning to evaluate data from the Kramnik mouse model. You know, the inventory that we've been able to collect is a substantial number of studies that examines incorporation of rifampin, incorporation of pyrazinamide, incorporation of moxifloxacin, replacement of rifampin with rifapentine, and use of wholly new drug combinations such as the PAMZ combination. And I've given you sort of the number of experiments of each type. These are all, again, relapse-based experiments that we have in this database. Some of these also include comparing daily therapy to intermittent therapy and of course, different durations of some therapies. So there's potentially a fairly robust collection of data that could be brought to bear. We're focusing first on the REMOX related trials, in part because that's where the most data lie, but also because of all of the other modeling efforts going on that you've heard about, in hopes that we'll eventually be able to integrate learnings across these models and become better able to predict these translations. So I don't want to, the statistical analysis plan has been developed in a preliminary way. I'm not going to spend a lot of time on this. Klaus would be delighted to talk ad nauseum on this. But I will just emphasize that we have developed a couple of potential analyses that we think can be applied to these types of data. Again, they are compromised to some extent because we have selected what is commonly used as a rather binary endpoint. Do they relapse or do they not in the mouse data, which may cause some challenges. We're certainly aware in the analysis. But for the more modern regimens, we do actually have multiple time points, which may allow some types of time to event analyses that can be useful, I think. So what I've hoped to impress upon you and to build on previous speakers is to say that we view this kind of analysis, what's already been done with the hollow fiber system, what we're proposing now with the sterilizing mouse model, as the initial steps to try and address this translational gap and to develop more confidence in these preclinical models. This has a variety of different advantages and is important in terms of defining what belongs on the critical path, where it belongs on the critical path, to hopefully increase the efficiency of interactions with regulatory agencies, and as I mentioned, to set a precedent that can be applied to other models that are in the preclinical drug development space. And I think perhaps most importantly, to identify what we still don't know and can't ferret out from data that are currently existing, and to define an appropriate research agenda to really try and fill in those gaps more completely. So with that, I'll stop, and again, just acknowledge the working group. We've already put in quite a few hours on these projects. Specifically recognize Nicole Ammerman, who's a new member of the group and is working on the data transfer at Hopkins. And I think that's probably the one point that I left out, is that we are, I didn't state it specifically, we are in the process of transferring all of the data from this, from the databases being kept by different researchers into the CPATH database. So we've developed a template for that. That process is now underway with the REMOX regimens, and as you might be expected, we are still refining that template to make that process as efficient as possible. So with that, I'll stop and see if there are any questions.